And remember that anytime you want, we do have those highlight reels, if you will, in Panopto as well. Those are, like I said, like 15 to 20 minutes, just kind of going over the high points of what to expect in the chapter. So again, if you haven't had a chance, or you forgot about those over the weekend, you should take a look at those because it gives you the big picture before we launch into the smaller details, okay? So those are available for you, and then, of course, these are being recorded and will be archived so you can go through them for later review as well. All right, so before we get into blood vessels though, let us do a little bit of review for cardiovascular. So what were two things that we think are really important or maybe a little bit detail oriented where we got questions upon them and we want to go over them from last lecture? So what are the two most important things that we were seeing last time when we were talking about cardiovascular physiology? I was like, I don't know, it was a vacation weekend. I proved that. I don't have to know it yet.
Layers of the heart.
Layers of the heart? Okay, so if we were talking about layers of the heart, how many layers of the heart do we have? 3 layers of the heart. So we've got an epicardium, which is on the outside. We have a myocardium that's on the inside. And then we have an endocardium that is on the very inside, right? So when I say myocardium on the inside, we're saying thickness of the wall. Remember when we were doing the sheep heart dissections last week? That's the thickest part of the wall. That's where all that cardiac muscle is going to be located. So endocardium is an epithelial lining to each of the different chambers. And we're going to see that same type of 3-layered organization when we get to blood vessels, or at least to the base blood vessels. So that's kind of a good transition to blood vessels because they have the same type of outer layer. They have a muscular middle layer, and then they have an inner epithelial layer, what we call endothelial layer. Because we kept saying that with the blood always is going to be interacting with epithelium. If it sees something different than epithelium, then that tends to start our whole chemical cascade in clotting, right? So if it sees collagen fibers from the outermost layer of a blood vessel, because it's breaking, the blood vessel is broken and now it's escaping, it's going to start that clotting cascade. If it escapes to surrounding tissues, then those surrounding tissues will also start the clotting cascade. You'll get clumps, clots, etc. So we see that in the inside of the heart too. We see an epithelial lining so it doesn't start clotting on the inside of the heart, okay? Now, if we're talking about layers of the heart, and we're thinking about lab practical, lecture exam, etc., remember that for the anatomy, we have a lot of anatomy that we tend to do in the lab, right? What's this, what's that, what's that? This week we're doing blood vessels, and it's a lot of where's this vein, where's that artery, etc. So if we're having anatomy questions, we'll have more anatomy questions in the lab than we will in lecture. You'll have some anatomy questions in lecture. So you're not going to see any on a lecture exam. It'd be very rare for you to see a diagram and it has a pointer that says, what's this layer like that, right? Because that's what we do in lab. So we try to parse it out in different ways. So, I mean, you could have like a multiple choice question saying, I don't know, of the layers of the heart, which one is made up of epithelium? And you have a multiple choice question that says tunica, or epicardium and myocardium and endocardium or whatever. So you could have, a question or two like that. There may be some anatomy questions on the lecture exam, to be sure, but we skew more in the lecture section towards physiology, where we're talking about how things actually work and function, etc. Because we have fewer questions like that in the lab. We have some, right, when we do tests, like blood tests, and we're going to do some cardiovascular tests next week, but it's a little bit more anatomy over there, okay? Okay, so another topic from last time.
Difference between the contractile cardiac cells and the autorhythmic cells.
Yeah, so those are both very important questions. So those relate to, when we were talking about physiology, we were looking at cells that gives rise to the tissues, gives rise to the organ, gives rise to the organ system, right? So cardiovascular system is about pumping and distributing blood around the body, getting what it needs out to it as well as back from it. And so we have vessels that we're talking about this time that are going to be the highways and roadways, which are going to be getting to the neighborhoods, which get deliveries to the individual houses, right? Like the Postal Service is going to be delivering to individual houses, picking up from individual houses. But there's main highways and even airplanes that are doing big deliveries, then it slows down and gets to smaller and smaller delivery pathways as we get to these individual neighborhoods. Of course, these days, right, Postal Service is not exactly the same. Now you have these big massive boxes, right? How many of you have an individual box at your house anymore? Rare, right? You have these big, like, you know, beehive type mailboxes where it handles like a whole, like 3 streets, and you have to walk to that one and get yours. But at least it's in your local area, and that's the idea of what we're seeing with blood vessels. So the heart, like we said, in terms of driving force for moving the blood around, it has 2 different types of cells that are making it up in terms of cardiac muscle cells. We have the 99%, which are the worker cells, which are the contractile cells, and then we have the 1%, which are the autorhythmic cells, which are more the management level cells. They're all cardiac muscle cells, but they have different jobs, right? The autorhythmic cells, very few, only 1% of the population, and they're only found in certain regions. They're found along the what? What system of the heart? There's a whole, it looks like wires. So the SA node is one part of it, and the pacemaker cells are making it the pacemaker of the heart. SA node, AV node, bundle branches, all that's part of the conduction system of the heart. Imagine, right, we have that diagram, you see those yellow pathways that look like nerves going through the heart. All throughout those yellow pathways, that's where we're finding the conductive cells of the heart, which are the autorhythmic cells. Those are the ones that are, as the name indicates, generating this regular rhythm, sending it out to the worker cells, telling them when to contract and when to relax, okay? So, when we were looking at that, we had some different pathways of routing cells. You saw I had more markers than just this one, but at least I have one. So we were looking at action potentials in these cells, and if we were comparing them, if we had what we had learned before, imagine if all these are labeled time and millivolts like that, we can write time like this, which corresponds to all these, some of that. So what we had previously in terms of like neurons and skeletal muscles, etc., is we had action potentials that look like this, big spike down, comes back up, and then flatlines again, right? So we had a resting potential, flatline, we had depolarization where it becomes less negative, repolarization where it becomes more negative, this one due to sodium flux, this one due to potassium flux. That's what we knew previously. Then we get to things like, let's say, autorhythmic cells. Autorhythmic cells don't have a resting potential, so they're constantly going to a depolarized state and then repolarizing, and then again, or the depolarized state quickly and then repolarizing again, etc. Okay? So we said that there certainly are some sodium channels that are contributing to this depolarization, but this big quick spike here is due to calcium instead of sodium. Meanwhile, the repolarization is still due to potassium, and we don't see a resting potential. When we look at the contractile cells of the cardiac muscle cells, we saw that these ones do have a resting potential like we see here, but then a big spike, and then this plateau before it comes back down again like that, okay? So in this case, what we had was sodium for the quick depolarization, potassium for the quick repolarization, but then calcium coming into play again and giving us this prolonged plateau period, this prolonged depolarization that gets the cardiac muscle cells a good long squeeze to be able to eject the blood out of the atria or the ventricles before we repolarize again. So we said they have a long contraction period. They also have a long, what we call refractory period, where they can't be stimulated again. With skeletal muscle, we can go bzz, 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 and we can stimulate it very quickly, and it generates more and more and more tension, or we can have little twitches or whatever. We got lots of different options. But for cardiac muscle, it's all about regularity instead. So we want to make sure we have long contraction periods, and we also have long, if you will, refractory periods, which lead to relaxation and filling periods. We want to reload again, and then do a strong, long contraction again, back and forth and back and forth. So certainly, knowing this changes the membrane potential of these different types of cells would be good. That's a big topic. Okay?
So first one is cell membrane potential?
This one here would be for like neurons, or skeletal muscle or something like that. So this is what we learned previously when we were doing 201. So this just gives us a comparison about what we knew before about excitable cells and then what it looks like now in terms of our excitable cardiac cell. Yeah. Okay. Okay, other questions? Yeah.
Intrinsic and extrinsic system.
Clotting?
Heart function. I guess I'm kind of confused of what those two.
Oh,
are.
so are you talking about like control and coordination
Yeah,
of cardiac
the-
muscle or activity?
Yeah, there's like an intrinsic system and an extrinsic.
Yes, so we're going to find that again and again, intrinsic and extrinsic control for these organs. Oftentimes with these different organ systems, cardiovascular is a good example, they've got a way of communicating and coordinating their activity internally, right? So this is like, again, if you're at your workplace, you usually have a way that you guys do things, you know how it runs specifically at your location, may be different from any other franchise or location, right? So you have your little internal communication system, your little intranet, or the way that you guys communicate. So these systems often have that as well, and that's what we're talking about in terms of the conduction system of the heart. They will generate a rhythm on their own. So the pacemaker of the heart and those autorhythmic cells will generate a rhythm of the heart that's like 70 to 80 beats per minute. So the SA node, the main controller for the heart rhythm, goes at about 70 or 80 beats per minute or so. Usually it's a little bit higher than that. But we also have extrinsic control coming from the outside, which oversees it and modifies it. So coming from the brain, right, we've got sympathetic and parasympathetic neurons coming down, which act like the gas pedal and the brake. So sympathetic stimulation of the heart is going to speed up our contraction rate. Parasympathetic is going to slow it down. So if we took both of those off, extrinsic control, the autorhythmic cells in the heart would still generate a rhythm on their own. You can take pacemaker cells from the heart, put them in a saline solution in a dish, just sitting there all together, and they will generate a rhythm all together, and they will start contracting on their own, okay? Without a brain telling them what to do, etc. So they have their own rhythm, but they have extrinsic control, sympathetic and parasympathetic neurons, which modify their own basic rhythm instead. So they speed it up and slow it down according to what's going on outside of the heart. Yeah?
What's the job of the slow calcium channels for that graph?
So the slow calcium channels, so what we find is that we've got some slow and fast calcium channels that are contributing to this depolarization here. See this area where it has kind of a less of a slope to it? So slow calcium channels are contributing in here, getting it to go to what we would call threshold. So remember we had threshold here as well at about negative 55 millivolts. So resting potential is about negative 70, and at threshold, if you depolarize enough to get the threshold, then you have this big spike that was occurring. So same thing that we see through here. So for instance, slow channels that we see through here are going to help the membrane potential get up the threshold, where then we're going to have these fast calcium channels that all open up almost simultaneously, give us this very steep slope to the depolarization over here. So it's kind of a finer point in terms of these ions moving across the membrane potential. Yeah. Okay. So we'll see that intrinsic and extrinsic controlled organs come up again and again. Like the digestive system has a way of communicating between the different organs of the digestive system, local control. Meanwhile, you've got extrinsic control, again, sympathetic and parasympathetic going down and communicating with it as well. One other thing before we get out of intrinsic control, when we're talking about the pacemaker of the heart, the SA node, then you have the AV node, then you have the bundle branches, etc. Each one of those actually has a different basic rhythm to it. SA node, like we said, is like 80 beats per minute. When you get down to the AV node, if the SA node goes out, if the main pacemaker of the heart goes out, you don't automatically die, right? What happens is the AV node takes over. It's like a backup system. You know when you have an electrical system and the generator goes out and everything kind of dims? You're still operating, but it's at a lower level. You're like, oh, the main generator went out, so we're having to rely on the backup generator. We still have lights, but we need to get that main generator going again, right? Same thing in terms of the pacemaker of the heart. If the SA node goes out, the AV node automatically takes over, keeps a rhythm of the heart of about 40 to 50 beats per minute until things can be put back into place again, like an external pacemaker be attached or whatever. So it keeps the body alive while that SA node is being not really repaired or in fact replaced with like an external one. Okay? Yeah.
Does sodium in the autorhythmic, is it where it's like starting to climb?
So sodium also contributes to the depolarization that we see over here as well.
Okay.
So there is a sodium component to the autorhythmic cells too. So both of the sodium and the calcium, they're mainly extrinsic. They're found outside of the cell. So when those channels open up, those positively charged ions flood into the cell, which make it less and less and less negative. So that's why whether you see the movement of sodium or the movement of calcium, they're both positive ions going into the cell, which is going to depolarize the cell. Remember that with potassium, like we see here on the other side, that's high inside of the cell. So when potassium channels open up, we're going to be losing those positively charged potassium ions out of the cell, which makes it more and more and more negative and returns it back to its state back before it gets stimulated. Okay? Aria's like, whoa, we talked about all that stuff before? It's a good thing we're talking about it again. All right. So take a look at that stuff. If that doesn't seem that familiar, do take a look at that stuff. Look at the highlight reel for the heart. I think we touch on those again. And of course, you can go back to the main lecture itself. You can go back in. You can type in autorhythmic cells into the little search window, and it'll take you right to that slide where we started talking about autorhythmic and contractile cells. And you can look at those charts again. Okay? Okay. So let's talk about blood vessels, which to a certain extent are a little bit easier than heart, a little bit. At the beginning of this, much like the heart chapter, we're going to start with anatomy. We're going to say what are the parts, and then we'll talk about what those parts do. Okay? So by the end of this chapter, we start talking about blood pressure and blood flow and distribution of blood, etc., which kind of sums up the entire cardiovascular system. We have the heart that's generating that rhythm, but it's these blood vessels which are helping to maintain the blood pressure and blood flow, distribute it to where it needs to go. Okay? Okay. So we're starting out with blood vessels as part of a closed system. So we mentioned this early on where we said that in larger organisms we have a closed system where mainly the blood stays within these blood vessels so that we can be assured that we have a distribution system that gets to each and every cell in the body. If you have a smaller organism, like a worm or something like that, they might have an open circulatory system, or it's more like a sprinkler system where we just like throw blood or hemolymph all over the internal organs, and they're small enough that they get what they need. Okay? But for us, we need a closed system of vessels that begins and ends at the heart. All right, so 3 major types of vessels. We're going to see these in lab, but of course we need to know about them here too. Now we say 3 main types: arteries, veins, and capillaries. There are small arteries called arterioles, and there are small veins called venules as well. That kind of gets us up to 5 different categories of blood vessels. Each one is different in terms of anatomy and physiology. They're going to be tied together. So we're going to see that their structures are going to be different from each other, and that's going to give them different functions. So we already know, right? Arteries take blood away from the heart. The A's match up. Veins take blood back to the heart. Likewise, small arteries called arterioles still going away. Veins coming back too. The capillaries are at the opposite side of the heart. The capillaries are the delivery point. So that's when we're kind of switching over between away and to. Okay? So capillaries are the things that are contacting the cells of the body and are at the sites of exchange. The other vessels are big enough, they're just about routing it to where it needs to go. So they tend to have thicker walls. We're not going to be exchanging things across the walls. We're going to wait until we get to the capillaries. So the capillaries are the most numerous. They're the smallest because they're everywhere, almost everywhere next to the cells in the body. There are some cells of the body that don't have direct blood flow to them. Remember when we looked at skin, there's epidermis and dermis. All those cells of the epidermis are relying on dermal blood vessels getting as close as possible to them, and then things just diffusing across the walls, okay? So there are some cells, even like cartilage, that don't have direct blood flow, but for the most part, capillaries are making contact with all the different cells of the body. So this is a typical schematic then of that whole cardiovascular loop. So we've already talked about this part up here, right? There's the pump. We have arteries that are going away. We have veins that are going back towards, and then this is the site of exchange, the capillary beds. So this is one other thing that we talk a bit about in lab today, that there is this behind-the-scenes series of vessels that are called lymphatic vessels that are a separate system from the cardiovascular vessels, but they work with the cardiovascular vessels. So they are in amongst the cardiovascular vessels, and they have a job of kind of being like the cleanup crew for the cardiovascular system. Because when we get to the capillaries here where it says exchange vessels, we're exchanging all kinds of things. We are dropping off respiratory gases, dropping off oxygen, picking up carbon dioxide. We're dropping off nutrients, we're picking up waste products. We're dropping in fluids to the tissues, and we're picking up fluids and anything that's kind of floating around in them. Okay? So if you get dehydrated, your tissues also get dehydrated. You can sometimes see it, right? People will get kind of skinny. You can actually see their skin doesn't have the resilience. It doesn't bounce back as much as it normally would, etc. So if we're going to rehydrate the tissues, we drink, it gets absorbed across the wall of the intestine, picked up by the blood, but now the blood has it. We're not fully hydrated yet. We've got to get it out to those cells that have all shrunken down. And so we need to get fluid out to them, but oftentimes we need to exchange it. We don't want to just constantly dump fluid out there. We want to exchange. So when that happens, oftentimes the amount that we dump out to the tissues is a little bit more than the amount that we get back. And so because it's not even like that, there's a little bit of fluid that gets left behind, and it's the job of these lymphatic capillaries to pick up this excess fluid that gets left behind and route it back towards the cardiovascular system. So what you can see there is that we have lymphatic capillaries too, but they're kind of blind-ended. They kind of look like little root hairs instead, okay? And they gather up that excess fluid. They route it back towards the heart. Notice that there are lymph nodes along the way. These are little filtration points, much like the fluid in your pool, if you will, right? You take out some of that fluid, you route it through a filter, clean it, and then you dump it back into circulation. That's what we're doing here. We're picking up that excess fluid, we're cleaning it along the way, and then we dump it back into circulation like that. Okay? So we'll talk about that as a separate system after exam 1, but it relates because it's part of this fluid system of the body. All right. So getting back to cardiovascular, though, arteries going away, capillaries, veins coming back. Now, the arteries, we have different types. We said there are arterioles, small arteries, but you can even see that there are 2 types of arteries that we see here, elastic arteries and muscular arteries. The deal here is that when we look at arteries compared to veins, the arteries are receiving the blood right from the heart. And the heart, due to these very powerful, almost violent contractions, are sending these big tidal waves of blood down the vessels on a regular basis, right? Unless you're dead. So you get about 70 milliliters of blood whooshing down these arteries all the time, and so the arteries have to be able to withstand that big tidal wave of blood without blowing apart. So they have a much thicker wall to them that we'll see in lab. Some of you have seen already. They have a much thicker wall compared to veins. And even with the arteries, there's 2 different types of arteries. The first ones that receive this tidal wave of blood first have a lot of elastic fibers in their wall so that they can stretch out and then do this elastic recoil and come back to their original size. The next ones, which are called muscular arteries, they still have some elastic fibers in them, but not as many. They have a more smooth muscle fibers in them, though. And what they do is they help to kind of route the blood towards these particular areas, and by constricting or dilating, they can help to kind of channel the blood where it starts to need to go, okay? Then we get to arterioles, and arterioles still have a muscle layer to them, but as you see there, they're called resistance vessels. The next vessel that we have after arterioles are really going to be our capillary beds. These are some kind of fine-tuning that we'll talk about in terms of blood vessels. But in short order, after these arterioles, we have capillaries. And capillaries are super duper thin and super small. Rather than having 3 layers like all these other blood vessels, they're a single layer of epithelial cells with a basement membrane attaching it. That's it. So we know that a single flattened layer of cells, right, that's like simple squamous epithelium. It's like the thinnest layer that you can get. And it's so thin that if we had this powerful high-pressure blood coming down to it without kind of reducing that pressure, then they would just blow out automatically. So these different arteries leading up to the capillary beds are helping to dampen the flow and reduce the pressure so that by the time we get to the capillaries, we've distributed it out to all these capillaries, but we've reduced that pressure so that they're not going to just blow up. Okay? So the arterioles, these ones here, are the last little protective vessel protecting the capillaries that are coming, and they're reducing the pressure and they're slowing down the blood flow so that by the time the blood gets to the capillaries, where we're doing this exchange, it's nice and smooth, it's not pulsing anymore, and it's nice and smooth and nice and slow. So we have time to do the exchange with the tissues before we then zip up to venules and veins and we're out again. Okay? And that's what we see on this side here. Capillaries go into small veins, which we call venules, which go into larger veins, which take it back to the heart. So you can imagine on this side over here, on the left side, the venule side and vein side, that's much lower blood pressure and blood flow compared to the arterial side, right? We're all the way around this loop on the other side, way far away from the heart. So, like I was talking about in lab a little bit earlier, you can imagine if you have a big dam or something like that, and we open up the dam and release some of that flow on a regular basis, kind of like the ventricles contracting. Right at the dam, you've got high pressure flow that's coming out, so you've got a lot of pressure you've got to deal with. And so as you get further downstream, though, as it goes from big rivers to smaller rivers down to streams and little babbling brooks, etc., that flow is going to be reduced. And so that's what we're seeing kind of around the other side. In fact, by the time we get to the other side, we actually have to help and push the blood to get it back towards the heart. And there's various ways that we're going to do that too. Okay? So we're dealing with 2 different things. On the arterial side, we're trying to like not blow up because there's so much pressure, and we've got a lot of anatomy that supports that. By the time we get to the venule side, we're trying to nudge it along and get it back towards the heart. Okay? Okay. So there, we're done. That's the whole cardiovascular system. We talked about it in one slide. You can just like flick through the rest of them on your own. You're good. All right. So let's take a look at the blood vessels in terms of that anatomy and physiology, right? So we said on the arterial side, seen on the left, we need to have reinforcements. It's high pressure, high flow. So we need to have reinforcements. So we have an extremely thick wall. So if you look at the thickness of this wall here compared to the thickness of that wall there, you can see that arteries have a much thicker wall comparatively. So if you compare the outer diameter, like from here to here and here to here, you can have, like the inferior vena cava and the abdominal aorta have the same outer diameter, but the thickness of the wall is going to be totally different. This will have a relatively thin wall compared to a relatively thick wall of the artery, okay? Because this one has to be able to resist that flow. So they both have 3 layers. They have a tunica intima or interna lining the inside of the blood vessel, just like we talked about endocardium. This is an epithelial lining to make sure the blood isn't going to freak out and go clotting on it. Then it's got a muscular middle layer called the tunica media, right, in the middle. This is smooth muscle instead of cardiac muscle. Cardiac muscle only in the heart. So now that we're out at blood vessels, this is smooth muscle. Then we have a tunica externa on the outside out there, and the tunica externa is going to be connective tissue. Okay? So it connects it to the surrounding other tissues. It also helps to hold it in. When we get to the capillaries, like we said, they also have an anatomy that helps their particular function. So in this case, what we have is there's that single flattened layer of epithelial cells with a thin basement membrane. So that is much easier to be able to allow things to cut across and distribute with the tissues. Okay? Now, one thing before we get out of this slide, this is another physical adaptation or anatomical adaptation that we have in the veins, valves. Okay? So what we find is that the innermost layers of the vein will form valves at regular intervals. It depends on the vein itself, but it can be like every centimeter or so, there's a valve. So just like in the heart, these are one-way valves. If the blood goes through one way towards the heart, it's okay. If it tries to slide back the other way, it fills those cusps and it brings them together and it closes the valve. So it's a bit like squeezing toothpaste, right? If you squeeze toothpaste anywhere in the middle of the tube, it exits one end and it has no choice. It can't go out the other end. So it doesn't matter where you squeeze it. If you squeeze it hard enough, it's always going to go out that way because it only has a choice to go that way. Same thing with these veins, right? Those valves ensure that regardless of which way the blood wants to go, it always has to go back towards the heart. Okay? So the best example thinking about that is if you think about like veins of the lower limbs, right? That's got to be tough to get that blood back from pooling in the feet and work it all the way back to the heart. So we've got these veins all the way along. So if we squeeze them, and in fact, the vein itself can squeeze itself. It has smooth muscle, so it can squeeze itself like a toothpaste tube squeezing itself and get that blood escorting back. But also veins tend to run through skeletal muscle. So when you're contracting skeletal muscle, it helps to squeeze those veins and escort the blood back further. Okay? So that's why when you're on like long flights or you're in the hospital and you're not moving a lot, they tell you about doing different exercises or getting up and moving every once in a while because it's going to squeeze that blood in the veins and get things moving along. Because like we said, when we were talking about blood, if the blood slows down too much, there's a chance it just gets bored. It's like, oh, what's around here? And I start clotting to the wall and form a thrombus or something like that, right? So we don't want that to happen. So the skeletal muscle actually helps to pump that blood along as well. Okay. So this slide just really goes through what we were talking about in terms of layers of the heart, right? And tunica interna or intima, we said that that's the innermost layer. It's endothelium, which is epithelium. This is continuous with the endocardium to make sure that the blood is always just seeing epithelium. So it keeps it nice. It's a slick surface. Make sure that it can move through easily, right? That's what we see with epithelium. A lot of our wet epithelial layers make it smooth or slippery. All right, tunica media, we said this is smooth muscle rather than cardiac muscle. And this is actually regulated by the sympathetic nervous system. So we'll talk about this towards the end, but we said that our different arteries and veins can constrict or dilate. They don't just do that on their own. They are coordinated by the autonomic nervous system. Now, whereas the heart had both gas pedal and brake, these ones are one of the exceptions to the rule in terms of what you learned about before. It was called dual reciprocal innervation, right? So when we were learning about autonomic nervous system, did you guys learn about that? Every once in a while you get people that come into the class, you're like, remember that from 201? They're like, nope. And then I'm like, all right, does that mean you don't remember it? Or like you didn't get taught it? They're just like, no, we don't have time to do that. We're skipping over autonomic. Sometimes that happens. My very first semester teaching 202 over at Southern Dobson, right? So I started the anatomy and physiology program over here. And so when we first started here, there weren't enough classes for me to teach. So I would like teach a Bio 160 over here, which is the intro course, and then I'd go over there and teach like a 202 or something. So I was teaching a 202 over there, and 202, like you've seen, starts with blood. The last thing that you guys did in 201 was nervous system and special senses. So I start up my spiel talking about blood and blood cells and stuff like that. I get this hand that comes up in the back, and they're like, so when are we going to learn about eyes and ears? And I'm like, that's not me. That was your last instructor. We got a whole bunch of stuff to do this semester. We don't got time to be doing catch up with that. So, point being, I'm hoping you've heard about autonomic nervous system. If you haven't, you want to review that. But autonomic nervous system, oftentimes with organs, has dual innervation, sympathetic and parasympathetic to each of these organs, which act like a gas pedal and a brake. In some cases, it's not exactly gas pedal and brake. They may switch. But most organs have both sympathetic and parasympathetic, and they have opposite effects. That's the reciprocal part. For blood vessels, they only have sympathetic. So sympathetic stimulation gets them to constrict, and if we want them to dilate, We just stop stimulating them, and then they can relax. So that's all we have. We have a gas pedal. So it's a bit like if you were driving a car and you were operating the car with just a gas pedal. All right, it is possible. It's not very precise, but it is possible to stop the car and slow down the car and speed up the car with just the gas pedal, right? You got to take your foot off the pedal and just wait for it to stop, but it'll stop, right? So same thing in terms of our blood vessels. So this smooth muscle stimulated to constrict by the sympathetic nervous system. When we want them to dilate, we're just going to stop stimulating it and they will dilate, okay? So we'll see that again later on. So tunica media, like we said, is going to be able to constrict and dilate, and that's going to change the diameter of the blood vessel. We're going to get to that as one of our kind of physiological parts of this chapter, where we're saying, oh, okay, so what's that going to do, right? Think of a hose or a straw where it has a bigger or smaller diameter. It makes a big difference in terms of blood flow and pressure, right? Imagine, if you will, you were at whatever, In-N-Out or something like that, and you get your drink, and you have one of those stirring coffee straws instead, right? Really, really small diameter. You're like trying to actually drink it, and you can't get it to go through very well because it has such a small diameter. There's a lot of resistance there, okay? So that also will happen with blood vessels. So by changing their diameter, we can change blood flow. We can actually change blood pressure in order to maintain it. Okay. So interna, media, and externa, right? Externa is also known as adventitia. This is where we said we have connective tissues. Specifically, we have some collagen fibers that are in there as well. Now, this isn't necessarily the most important thing, but interestingly enough, some of your blood vessels are so big that you will have a series of blood vessels that will be blood vessels to the blood vessels. So I thought maybe they had them in this diagram over here, but they don't seem to. But this thing that we see here, the vasa vasorum, most blood vessels, right? A blood vessel technically counts as an organ, right? So if we say this is an organ system, this is, right, cardiovascular system. A system is made up of a bunch of organs working together for a particular function. So we're like, name the organs of the cardiovascular system. You're like, heart, and then you're like, oh, right? So each and every artery, vein, arteriole, and venule counts as an organ because by definition, an organ is where we have more than one tissue working together for a function. And we just went through at least 3, if not 4, different tissues, right? Connective tissue, muscular tissue, epithelial tissue, and then we said nervous tissue comes down and controls these as well, right? So these different organs actually need their own blood supply, right? They need their own oxygen to the smooth muscle cells so that they can survive. They need to get rid of waste products, all kinds of things like that. Usually, they're just siphoning them right off of the blood that's passing through them. But when you get really big blood vessels, like the vena cava and the aorta and stuff like that, the wall is so thick that those cells that are right in the middle of the wall, they don't have any possibility of allowing that stuff to get to them. So what they need is an additional blood vessels. There are blood vessels that are on the surface of the blood vessel that are feeding it to make sure it's alive. So much like the coronary circulation feeds the wall of the heart, its own little circulation, sometimes with very big blood vessels, you'll get a little circulation and little blood vessels that are feeding the wall of it. Okay? So that's what that means. It's smaller blood vessel network that are supplying these larger blood vessels themselves. So again, not critical for the exam or anything like that, but it's interesting when we're talking about the needs of these different organs that some of these blood vessels, right, are big enough that they have to have their own blood supply. Okay. So that's the big picture for vascular component. Questions so far?
The bumps there on where for the veins, or is that?
These bumps?
No,
Those bumps.
Yeah, are those where
The valves are?
the valves are located?
Yeah. So they're trying to show you where some of those valves are located. Valves will be located all the way through as well. Yeah. So even up to the larger veins as well. So vena cava and stuff like that, you'll find them in brachiocephalic veins, things like that.
And working against gravity by restricting.
Yep, so they're always ensuring that the blood that's flowing through the veins is always working its way back towards the heart. Yeah.
So like, varicose veins, is that when the valves stop working?
Right, exactly. So we'll see that. What we're doing now is we're going to do a more detailed tour, and we're going to look at each of these different blood vessels in turn. Okay? So when we're talking about, when we get down to veins, we'll be talking about, okay, how veins work and routing this blood back to the heart, and how they're supposed to have these valves that are always functioning to make sure it's a one-way flow. But through time, everything breaks down, right? So, in fact, with these veins, particularly ones that are way down the lower poles of the body that have gravity working against them all the time, particularly in people that spend a lot of time on their feet, sometimes these things will break down. And so the valves that are supposed to be escorting that blood back, they start breaking down. Now you have less valves there. That means there's more of a chance for blood to pool in these veins located in these areas. And so with varicose veins, the valves are breaking down. So there's more blood sitting in them. More blood, it has to make room for more blood. So they tend to expand a bit, both in terms of diameter, and they even expand in terms of length. So you see them getting this kind of sinewy look to them as opposed to being just one long straight channel before, because they have to start accommodating that. There's different types of treatments for that as well. None of them are particularly pleasant. One of the things, they go in there with like a ramrod and they ram it through there and they create scar tissue in there, which automatically narrows the vessel, which helps to keep it going back. But, you know. Okay. So let's start talking about arteries. So arteries on our tour are all routing the blood away from the heart. So we said that there are two main types of big arteries: elastic arteries, which come first, which have to be able to stretch out when that tidal wave of blood comes, and then muscular arteries after. So the aorta and its major branches are going to be elastic arteries. So those in the morning lab, they know some already, right? So what are some of the big branches that you've seen in terms of big artery or arterial branches? So brachiocephalic artery is a good one, right? So you have the artery of the aorta, you have brachiocephalic artery, you got left common carotid artery, and you got left subclavian artery. All of them going to be elastic, right? You go down, thoracic aorta, abdominal aorta, and then you have some big branches coming off of there, right? What's the first big branch? It's a trunk. Celiac trunk. Celiac trunk comes off, you got an artery that goes to the liver and to the spleen and to the stomach, right? So celiac trunk is going to be a big elastic artery as well. Superior and inferior mesenteric, big elastic arteries, etc. Okay? All right, so elastic ones have more elastic fibers in them, and they tend to have a larger lumen to them. We want to, big tidal wave of blood, let's just spread it out. Let's get it to these different organs as fast as we can. So we don't want to have a lot of resistance. We want to dial these blood vessels open, but we want to make sure that as this tidal wave of blood comes down, we want to be able to expand like a balloon quickly, but then have this elastic recoil. So we have elastic or elastin fibers throughout the layers of these walls. So that way, we can withstand and smooth out these large blood pressure fluctuations. So later on, like next week, and even maybe at the end of this week, we'll talk about when you're measuring blood pressure. First off, you're measuring blood pressure in arteries, right? When you're doing blood pressure, you're not doing it in a vein. You're doing it in arteries, and there's always these two different numbers, right? So it's like, whatever, 120 over 70 or something. Why is there two? Because tidal waves of blood coming down. So when the ventricles contract, whoosh, there's a big tidal wave. So the artery's going like this, it's expanding out, high pressure right then, tidal wave passes, now elastic recoil, now it's lower blood pressure again. So it goes back and forth all the time. So it gets this pulsing. So when you're taking a pulse, which we'll do next week as well, always doing those on arteries. You try to take a pulse on a vein, you're going to guess that your patient's dead. You're going to start shocking them, they're going to be yelling at you not to, etc. Okay? So arteries do this pulsing like that. That's what you're feeling, is you're feeling those tidal waves. Every time you feel it come up against your fingers, that's when the ventricles are contracting and that tidal wave is reaching out there. Okay? Okay. So they do this elastic expansion, and they don't blow out, and then they do this rubber band snapback because of the elastic fibers. So if you think about it, when the ventricles are contracting, now we've got this big 70 mils that we're pushing out again. So high pressure, but what happens when the ventricles are relaxing and filling when they go into diastole? So, what's that? It's resting, exactly. So the heart's resting. We're not jacking any more blood out of those arteries. So does that mean the blood flow just stops? Like it goes, whoa, and then it stops, and then it goes, whoa, and then it stops? Like the way your mom drives your car that drives you crazy? No, it's a continuous flow. And so what happens is the first big push comes from the ventricles contracting. That's systolic pressure, the high point, like the 120. Then, when these walls of these elastic arteries snap back, then that produces an additional blood flow. So they balloon out, there's blood out here now as the artery is stretched. Then there's an elastic snapback. All this blood we push down. So there's no new blood coming in, but there's this elastic recoil of blood in the arteries which we're continuing to push down. So downstream, it sees a difference in terms of blood pressure, but blood flow is continuing to happen. So in that way, these arteries are often called pressure reservoirs, right? A reservoir where you can store something. When they balloon out like that, when the ventricles are up there relaxing, we get this elastic recoil. They almost act like a secondary heart. They are actually pushing blood again down. That's expanded their walls temporarily. So you can see that. We usually have a diagram of that coming up. Okay? All right, so we've got first our elastic arteries, then our muscular arteries. So the muscular arteries are distal, right, further out at a distance from our elastic arteries, and these are going to the specific organs. So they are starting to be the specific branches. Like we said, as we get down to hepatic artery and gastric artery and stuff like that, we're getting more towards our muscular arteries. And so these have a thicker middle layer with more smooth muscle, and they can then actively route blood to and from these different organs. They're not going to cut it off. They're not going to cinch themselves off, but we do know at different times you need blood to go different places. So, for instance, if you have a big meal, right, Thanksgiving coming up, what happens after you have your big meal? You take a nap, right? So, it's called postprandial somnolescence. It's got a fancy name, right? Everything has a fancy name. So, when you're tired, right, what your body's doing is, you're like, okay, I got a whole load of food here. I got a lot of muscular contractions I got to work on. I got to absorb things. I got to move it on further. I need a lot of blood going right through here. So we're going to take it from other places. We only got 5 liters, so we're going to route it to where we need it most. We're going to take it from other places. We're not going to cut them off, but we're going to constrict to these other organs and route it and dilate it to these important ones at the time. Okay? So these muscular arteries help to do that. They help with vasoconstriction and vasomotion by either increasing the blood there or decreasing the blood there, depending on the needs of the body and the organ at that time. Okay? Same thing in terms of, if you're running, right? If you're running, we need a lot of blood going to those muscles of the lower limb. Maybe not as much to the upper limb, lower limb for sure. Don't need as much going to the digestive organs when you're running. Let's hold off on that, route it to where we need it. Okay? So it all depends. Okay, so muscular arteries are the next ones. Then from muscular arteries, we go down to arterioles. And arterioles are our smallest arteries that lead to the capillary beds, and they are also about control. Okay? They're controlling the flow into the capillary beds, again, with that what's called vasomotion, vasodilation, and constriction. And remember, when we see vaso here, bless you, most people look at vaso and they think vein. Vaso doesn't mean vein, it means vessel. So this is constriction and dilation of vessels. So it can happen in any blood vessel that has smooth muscle in the middle of the wall. So that basically means any vessel, not the capillary. So these arterioles are the last, if you will, line of protection for those capillaries. So they want to protect them by slowing down the blood and taking away that whole pulsing sensation that we saw with the elastic arteries and the muscular arteries up above. Because if we have those big pulsing sensations of the blood, when it gets to the capillary, they'll just blow apart. Okay? So the arterioles do a good job of taking away that pulse and making sure that the blood is a nice, smooth, slow, laminar flow when it gets down to the capillaries. So if you look at their structure between the elastic artery, the muscular artery, and an arteriole, you can see how their anatomy changes based on their physiology, right? So look at this. Endothelium, elastic tissue, smooth muscle tissue, and then connective tissue, right? If we look at the elastic arteries, the first ones, they have the most elastic tissues in them because they need to be able to stretch and do this elastic recoil, okay? When we get down to the muscular arteries, we can see they still have connective tissue, but they have much more smooth muscle to them. Their wall is much thicker so that they can control the flow to the different organs. When we get down to the arterioles, overall, their wall is getting thinner, okay? But we still, of all the layers, we still have the highest amount of smooth muscle in them, so we can control the blood flow going down to the capillaries, okay? So their anatomy is changing associated with their physiology according to what they have to do. Okay. So questions on any type of arteries that we're looking at so far? Elastic, muscular, arterioles. So thinking forward in terms of the exam, you could have multiple choice questions where maybe it's a flow question, and so they have different vessels, and you have to find out the right arrangement. That, you know, elastic come before muscular, come before arterioles, come before capillaries, and they're in mixed up orders. You could talk about, you know, like we said, like the thickness of the walls, which one has the most elastic fibers, and then you see different blood vessels listed out, and you know elastic arteries are going to have the most elastic fibers, things like that. You could have, this could be, it probably is on our top 10 list. When we talk about short answer questions, this is a good short answer question because it's like we got all these different types of blood vessels. We can compare and contrast them. So like name 4 different types of blood vessels and tell me how their structure relates to their function, right? Very thin walls, very thick, smooth muscle layer, a lot of elastic fibers or whatever, and then why does it have that? Now, again, if we are talking about short answer questions, I usually give you a choice about what to tell me, right? So we have already named 3 different types of arteries: elastic arteries, muscular arteries, or arterioles. Then you have capillaries, then you have venules, then you have veins. So lots of different types of blood vessels. If I ask you 4, then it's your choice as to which ones you tell me. You can tell me arteries and veins and capillaries and then pick one other of the other subtypes or something. Okay? All right. So, capillaries. Smallest blood vessels, walls are super duper thin, right? So they basically have that smooth muscle layer, and that's it. There's a little basement membrane underneath it, but that's all they have is the thin epithelial layer. Their overall diameter is so small, like we mentioned when we were talking about blood, that a single red blood cell is only able to pass through at a time. So oftentimes, maybe on Thursday, if you remind me in lab, or you can certainly look it up yourself, it's always interesting because in certain organisms, you can actually see their blood vessels, right? So in very pale, kids or something like that, no, not that one. But like in other types of organisms like goldfish, interestingly enough, in their fins, right, their fins are living tissues, but oftentimes the fins of fish are clear and they're very thin because they need to be very flexible, etc. So the blood vessels that go through them are some of the smallest ones, but you can actually see them from the outside. And so what you can do, what we've done in the past, but you can see it in a video, is you can get a goldfish, don't worry, it doesn't die, and you can very gently fold it into a wet cotton swab, like a little wet sleeping bag, and put it on the microscope. And if you use the medium power objective, right, the low power objective, you can actually see the blood vessels in the fins, and you can see the blood cells going through all of the different blood vessels, just single file going through those capillaries and stuff like that. So it's pretty cool. On Thursday, if you remind me, I can at least find a video for you and show you how it looks in class. But the capillaries are small enough to where, like we said, the important thing is they are found almost everywhere, and they are next to almost every single cell of the body. So that way they are serving them to exchange these materials between them. Okay. So when we look at these, what we find is, even with the capillaries, there are actually different structural types of capillaries based on how much exchange we need. So when we look at these capillaries, one of the other things that's interesting to and specific to them is that there are these specialized cells that are called pericytes, and these pericytes help to control how much material is able to get across. So they stabilize the capillary walls, they control permeability, and they may also help these capillaries to repair. So there's a couple of interesting things about that. Number one, when we talk about exchange, it's not a standard rate of exchange. In fact, these epithelial cells, they can kind of spread apart from one another, creating big gaps in between, which obviously makes it easier for things to get across. Or we can have them butt up against each other very tightly, and then things can still get across, but not as easily. Okay? So the pericytes that are around them help to kind of shift, like taking plates or something like that and shifting them apart or pulling them together. They help to regulate how leaky these capillaries are. So that's one interesting thing about them, to be sure. In terms of vessel repair, the capillaries are delicate enough that we can actually build and destroy capillaries as need be. So for instance, if you start to get tissues that are more active and they need more blood flow, it's very hard for your body to construct a whole new artery, right? 3 layers and all that type of thing, etc. But capillaries are really easy. And so if you start increasing the demand of a tissue, you can actually grow more capillary beds, much like you talk about the nervous network, where you have dendrites that can make more neural connections and create a more complex neural web. Same thing in terms of capillary beds. So if we start developing more cells in these tissues, or the cells get bigger, right? You start going to the gym. You don't have time for that. You're in school now, right? But you know that gym where you pay the membership every month where you don't go? If you were to go there and follow your money, where it's going, then if your muscles get bigger because you're putting an increased demand on them, muscle cells, as they get bigger, there's not more muscle cells, but they're actually gaining, if you will, girth. They're getting bigger in terms of their diameter, more actin and myosin, et cetera, inside of them, right? More proteins in there, more activity, we need more oxygen, so you actually grow more capillary beds feeding those cells as they get bigger. So you can create more capillary beds. Now on the flip side of that, if you're not going to the gym and you're growing in other ways, yes, we're growing more tissue. Adipose tissue is living tissue. Those cells need oxygen and all that type of thing, right? So when you actually grow more fat, if you get to the point where you need more adipose cells, then there's a whole bunch of other capillaries that grow with it. So if you're gaining weight, there's 2 levels of gaining weight. One is the fat cells that you have, they just get bigger. Okay? You store more triglycerides in them, and they go from being small to being big. So you can have weight gain and weight loss just with the same cells getting bigger or smaller, kind of like when we talk about fluid. But if you go to extreme levels, right, like in your freshman year, freshman 20 or whatever it's called now, 40 or whatever. So if you go to extreme levels where you are getting much more tissue, much more energy that needs to be stored, then those adipocytes, those fat cells, are doing their darndest to get as big as possible, but that's not enough. You start creating more fat cells, and those fat cells have to be served, so you get more capillaries serving them. Then when you lose weight, hopefully, junior year, senior year, when you're like, I got to go get a job, I got to get rid of this. So then at that point, those fat cells, you're going to be shrinking them, but in fact, after you create fat cells, you can't get rid of them. You can shrink them, you can make them smaller, but once you've made fat cells, they basically hang around. So the only way you're going to get rid of fat cells in totality, that type of thing, right? So liposuction, getting rid of the actual cells, is the only way to get rid of them if you get that much bigger. So you have to, they can shrink, and you can certainly lose weight, but like you see in like the extreme person makeover, whatever those are called, right? They're usually doing some surgery. They lose a lot of weight, to be sure, but in order to get rid of that excess tissue that's much smaller, they have to do surgeries as well. Yeah? Cellulite. Mm-hmm. So cellulite, right, that has to do with, there's connective tissue that's kind of holding that together, areolar connective tissue and stuff like that that's holding it down in particular places. And that's where you kind of get some of the dips. But then it's the adipocytes, or the fat cells, that are expanding in the other areas, which gives you that kind of uneven texture. Yeah. Which, I mean, obviously you don't have to be that big to have cellulite. Everybody's got fat, right? Subcutaneous fat, etc., as well as fat in amongst the organs. Fat on the heart, right? You saw the fat on the heart. There's fat everywhere. Fat's necessary as energy storage. It's just you don't want too much of a good thing, right? Yeah.
So that's why when you have multiple pregnancies and you have lots of that, that's why so they just pump up?
Yeah, sure. So when you have pregnancies and stuff like that, you're supporting 2 individuals. You need more reserves. In fact, you grow a whole bunch more blood vessels during that as well. And sometimes those blood vessels break because there's blood vessel and blood pressure differences during pregnancy. Your blood volume goes up because you're serving the baby. We've seen that, right? You've seen there's the placenta there. That's a whole other organ that you've grown that you have to support while you're pregnant, right? There's the blood. All that baby's blood, all those baby's tissues need to be supported through the maternal circulation. So everything gets bigger. You get extra energy reserves. And so those energy reserves, when the baby's gone, right, you lose that weight from the baby and the placenta, and you lose some of this other weight, but those fat cells that you've generated, those tend to hang around. And so you can make them smaller, but it's harder to actually, you can't really get rid of them. Yeah. So, what are we talking about? I don't know. Blood vessels? Yes, blood vessels. So, capillaries are simple enough blood vessels that we can actually create new blood vessels and get rid of blood vessels if we need to. So they are simple enough that we can create them and destroy them as necessary. And these parasites, right, parasites not like eating you, it's spelled differently. These cells that are around our capillaries help in this process in terms of maintaining them and also affecting their permeability. Okay. So, like we said, these capillaries, as well as their surrounding parasites, are basically almost everywhere. All right, so even with the capillaries, though, we have different types of capillaries that are doing their function of exchanging with tissues, but they are better or worse at it depending on the needs of the particular organ or system or tissues. So these ones are the ones you typically think of. These are called continuous capillaries, and if you look at it, these are the simple squamous cells, right? Here's the basement membrane underneath, and everything looks like you would expect, like we've drawn out previously. Now here we see gaps, right? These are intercellular clefts. This is where some exchange occurs. Some exchange goes right across the wall of these flattened cells, okay? So what we find is our muscle generally gets served with this, connective tissue, the brain, nervous tissue, respiratory tissue. A lot of the tissues in the body have this type of capillary. Allows nice amount of exchange. But if we need even more exchange, then we get to fenestrated capillaries. So fenestrated capillaries, fenestrate and fenetra. So if you look at the Latin base, and if you look at even like some of the Latin-based languages like French or whatever, so if you look at window in French, it's fenetra. And so fenetra is a window, and so these capillaries have small windows in their epithelial cells. So you see all those little holes? They actually have more holes, and so that makes it easier for things to get across. In fact, there's a verb that's not very well used. It's called defenestrate. So that's your big word of the day. So if you throw something out of the window, that's called defenestration. So like those rare people that still smoke, right? And then when they get done smoking, and they go bink out the window, and you're like, what the? Really? I mean, like if they finish up their burger or whatever, they wouldn't be like, oh, bink, right? But somehow the cigarette butt, they're like, no, I think it's biodegradable. It's totally okay, right? No. In any case, right? These are windows inside of these epithelial cells here, so they make them leakier. So if we look at it, kidneys, small intestine, choroid plexuses, endocrine glands. These are all organs where there's a lot of movement going on. Kidneys are doing conditioning on the blood, right? They're making sure the blood volume, the blood concentration is okay, so there's stuff going back and forth all the time. Small intestine, a lot of absorption going on there. All of your nutrients are coming across in the small intestine. Choroid plexus, what's that? You're like, it's kind of... Yeah, right? Cerebrospinal fluid, we talked about that before, in and around the brain and spinal cord. These are the networks where that cerebrospinal fluid is created. So we got to have a lot of flow there. And then endocrine glands, what do those do? Right, so they're creating hormones and they're dumping them into the blood. Hormones are fairly big molecules. it's not as easy to get those across the epithelial cells, so we need some extra holes, okay? The last type, after the fenestrated, are called sinusoids, right? A sinus is like a big space. Sinusoids is a type of capillary. They basically look like they're moth-eaten, right? They're barely holding together. Not only do they have large holes in the epithelium, but even the basement membrane has holes in it. So these are where we have to have a lot of movement. Again, we're seeing for organs that put out a lot of, endocrine organs that put out a lot of hormones, for sure. The spleen, why does the spleen relate to the cardiovascular system? So when the red blood cells wear out, the liver and the spleen are one of the places where we take them out of circulation, right? So we have big macrophages there that take them out of circulation. So these are the places where we actually have cells, not molecules that we need to cross the wall, but whole cells that we need to get across the wall. And so we need to have big holes for that. And likewise, bone marrow. Why is bone marrow important? That's where red blood cells are produced, white blood cells produced, and platelets. They all have to get into the blood vessels. So we need to have big holes to allow them to do that. Okay? So these are, yet again, 3 more different vessel types that you could use as examples for comparing and contrasting blood vessels. Okay? So lots of different types. All right. So that's our first part of our tour for blood vessels. We made it down to capillaries in terms of our tour. We'll work up to venules and veins, and then we get into physiology, function, about how these things are moving across, etc.